Good morning, everyone. My name is Jenny Walters, and I'm a fifth-year graduate student in Dr. Hee-Jung Chung's lab. My MBM co-advisors are both Dr. Chung as well as Dr. Hee-Jun Kang. And today I'm going to be talking to you all about my research focusing on the pharmacological inhibition of a brain-specific tyrosine phosphatase called STEP and its role in reducing KA-induced seizure propensity and hippocampal network excitability. Our lab studies the underlying mechanisms of epilepsy. And first and foremost, it's important to note that anti-epileptic drugs, or AEDs, do not work for everyone with an epilepsy disorder. There are various kinds of AEDs that are on the market, with some of them with multiple mechanisms of action. However, still for certain patients, such as those that have temporal lobe epilepsy, or TLE, where they have seizures that originate within the hippocampus, and then slowly spread out through the cortex to the rest of the brain. Unfortunately, about 1/3 of these patients go on to develop drug-resistant seizures. So really finding small novel chemical compounds that could be potentially used as an AED are in critical need. And this is what really led me into the researching the compound TC2153, which is an acute STEP protein inhibitor. So TC2153 ultimately creates an irreversible covalent disulfide bond within the catalytic region of the STEP protein, thus rendering it inactive. So I'm sure you might be wondering, why is STEP protein so important to us, or why is it so significant in the case of epilepsy? Well, the upregulation of STEP protein expression is highly associated with various kinds of neurological disorders, including but not limited to Alzheimer's disease, schizophrenia, Fragile X syndrome, and epileptogenesis. So really what we wanted to know was, will using an acute STEP inhibitor such as TC2153, will it have any effect in an in vivo model on seizure propensity or severity? And we recently published a paper in Epilepsia earlier this spring, myself as well as our postdoc in the lab, investigating this question. So first we looked at whether TC2153 has any effect on seizure susceptibility. And what we did was we used a wild-type model of mice bred on a C57BL6J background. And we first administered the TC2153 intraperitoneally into the mouse, allowed it to circulate for 3 hours, and then injected the same mouse with a chemoconvulsant called kainic acid. And what kainic acid is it's a glutamate analog that will bind to kainate receptors that are highly expressed within the hippocampus. And when it binds, the receptor, the channel opens, causing or allowing the positive influx of ions into the cell, causing the cell to become hyperexcitable. And this ultimately induces the seizure phenotype that we can visually assess in this model. So we broke our data out. We looked at both male and female cohorts. And as we can see here, in the male data, there are several time intervals where there's a significant reduction in seizure propensity in the mice that were treated with the TC2153 compound prior to being injected with the chemoconvulsant. However, when we look at females, the female graph, we saw an even further reduction in seizure scores across, I should have mentioned, across a 120-minute time period following the kainic acid injection. So not only do we see a further reduction in seizure severity, but also we see it at nearly every time interval. So this led us to wonder, well, what could be causing this difference between the female seizure susceptibility and the males? And really what we came to was, well, maybe there's something specific about the female physiology that is interacting with this TC2153 that's allowing these effects to occur. So our following experiment looked at the same wild-type model of mouse. However, they were ovariectomized, and this ultimately just means that the ovaries were removed, and therefore this eliminated the circulating estrogen and progesterone from the system of these female mice. We performed the same procedure where we injected the TC2153, and then 3 hours later injected with the chemoconvulsant kainic acid. And we found that in the ovariectomized female mice, that this TC2153 induced reduction in seizure severity was ultimately rescued and brought back to near baseline levels, as, similar to the vehicle injected mice. So what this ultimately showed to us was that there seems to be some type of interplay with female sex hormone, whether it's progesterone or estrogen, and TC2153 to cause these effects. Now, as part of a further project down the line, we would be very interested in looking at hormonal therapies in this same mouse model to see whether estrogen, progesterone, or perhaps both hormones are responsible for these effects. Next, we also wanted to look at the effects of TC2153 on hippocampal excitability. So what we did was we took a acute hippocampal slice, and actually, as Dr. Woodward kind of talked about in his previous talk, we used GCAM success mice. So essentially, this mouse line expresses a genetically encoded calcium indicator called GCAM success, in which when there's a positive influx of ions into the cell, this causes a conformation change in the protein, pushes the fluorophore up for the surface of the cell, and it fluoresces, and we can measure the intensity and thus analyze the excitation of these cells within each hippocampal region. So we looked specifically at the dentate, CA1 and CA3 regions. And just to describe the methodology a little bit, we first placed the slice in an ACSF bath or artificial cerebrospinal fluid. We measured the basal level intensity, and then we introduced 15 milliliters of potassium cyanide to the slice for 2 minutes in order to excite the cells. And you can see here in the representative images, one set of slices was treated with DMSO control, and then one set of slices was treated with TC2153. And we also broke this data out by male and female cohorts. And what we found was that specifically within the dentate and CA1 region, so both males and females, there was a significant reduction in fluorescence intensity that was measured in slices that were treated with TC2153 compared to the DMSO treated slice, as well as a decreased trend in the CA3 region. So what are our future goals for this project? Ultimately, we want to see if TC2153 exhibits translatable effects in human models, such as human iPSC-derived neurons. We're already in the process of addressing this question in our collaboration with Dr. Jeff Parent from the University of Michigan, who has kindly sent us some of our own to model. And I also, during the spring semester, completed a traineeship in Dr. Hugh Jun Kong's lab looking at mouse neural stem cells. And we actually gathered some exciting data looking at the treatment of these cells under conditions either with treatment of DMSO vehicle control or TC2153. So in the process of analyzing that, so I'm excited to see the results. And then I would just like to acknowledge my lab, the Chung Lab, specifically my PI, Dr. Kijun Chung, as well as our postdoc, Dr. Ying Kim, and the graduate students in our lab, and as well as undergraduate mentoring. And then I would also like to thank our grant funders and, of course, the MBM program for being an immense source of opportunities and as well as funding this research for the last 2 years. So thank you very much.
Questions? Oh, yeah, Roger?
Sure, I have one. So I'm wondering how you're going to go about the experiments that you're planning to do with the iPS-derived neurons. Will that be in 2D, 3D? What kind of measurements do you make, and how do you compare that with your mouse studies?
Right. So so the traineeship, basically, I was being trained how to grow and maintain the mouse stem cell line and differentiate them into neurons or astrocytes. So basically with the human iPSC derived neurons, we would probably be performing something similar where we look at perhaps using a calcium indicating dye and looking at both sets of neurons under the conditions of either being treated with the DMSO control or TC2153, and then performing the calcium imaging to see how that affects the fluorescence intensity. Thank you.
So do you also incorporate some kind of hormonal stimulation into those cell cultures?
So that would be really interesting, and we've actually talked about doing that. So I think that is an avenue that should definitely be pursued. Is either treating the dishes, you mean with like either estrogen or progesterone or both to see what the effects are on the activity. Yeah. Definitely.
We have a question from Chad, from Robert Frenchick.
Mhm.
Besides looking at an increase of calcium, did you notice a change in calcium oscillation frequency?
Oh, that's a good question. I did.
Please speak up.
Oh, sorry. Yes, that's a good question. I did not notice that or analyze that, but that would be really interesting to look back at to analyze. Yeah. Mhm.
Any other questions?
Thank you.
Jenny.